Hello guys, today we will continue our lecture about the nervous system. We know that there is four basic tissues in the body: connective tissue, epithelial tissue, muscular tissue, and nervous tissue. Nervous system anatomically is formed from CNS, which is the brain and spinal cord, and PNS, which is the peripheral nervous system, consists from nerves and associated ganglia. Histologically, it formed from nerve cells, which is called the neurons, and glial cells, which is called the neuroglia. The last lecture, we talked about nerve cells. Today, we will talk about nerve ganglia and nerve or neuroglia. Nerve ganglia. It's a collection of nerve cells and fibers outside the CNS, covered by connective tissue sheaths. So there is no nerve ganglia inside the CNS. Nerve ganglia presented only in the peripheral nervous system, like that. This is a part of spinal cord, which is a part of CNS, and this is peripheral nerves. So this is ganglia presented in the peripheral and not presented in the CNS. Again, nerve ganglia presented only in the peripheral nervous system. There are two types of the nerve ganglia. Nerve ganglia may be spinal and may be autonomic. What is the difference? Spinal ganglia presented in the cranial nerves 5 and 7 and 9 and 10, and also in the dorsal root ganglia, while autonomic presented in sympathetic and parasympathetic ganglia. Capsule of spinal ganglia is thick and autonomic is thin. The nerve cells presented in spinal ganglia is pseudo-unipolar, which is few, variable in size, arranged in groups, and has a single T-shaped process which convoluted forming glomerulus, while that of the autonomic ganglia is a multipolar cells, large in size and equal, distributed in all over the ganglia with no glomerulus. Glomerulus presented only in the spinal ganglia. The nerve fibers in the spinal ganglia is thick and myelinated, while that the autonomic is thin and non-myelinated. Synapse. There is no synapse in the spinal ganglia and there is synapse in the autonomic ganglia. What is the synapse? Synapse means connection between two nerves or connection between nerve and muscle. Spinal ganglia, there is no synapse between these nerves, while autonomic ganglia, there is a synapse between these nerves. The function of a spinal ganglia is mainly sensory, and the function is autonomic ganglia is mainly motor. So again, this is a very important table. Spinal ganglia presented in the cranial nerves and spinal dorsal root ganglion. It's a thick with pseudo-unipolar nerve cells that forms a glomerulus with thick and myelinated nerve fibers with no synapse which have a sensory function, while autonomic presented in sympathetic and parasympathetic ganglia with thin capsule, multipolar nerve cells that forms no glomerulus with nerve fibers which thin and non-myelinated. Synapse between them is presented with a mainly motor function. This is the spinal ganglia with thick capsule, and this is a thin capsule. The fibers here is pseudo-unipolar cells, and this is a multipolar cells. There is a myelin, there is myelin, there is also myelin. This is the spinal ganglia stained with hematoxylin and eosin, contain cell bodies of the pseudo-unipolar nerve cells like that. In between there is a satellite cell, which is the cell type of the neuroglial cell presented in the nervous system. This is the spinal ganglia stained by silver stain. This is circles is the cell bodies of the unipolar nerve cells. This is the sympathetic ganglia stained with hematoxylin and eosin. Presents the cell body with multipolar cells, and in between there is a satellite cells which is a neuroglial supporting cells. This is a thin capsule of the sympathetic ganglia, and this is a fat cells. This is a sympathetic ganglia stained especially by silver stain. So the special stain of the ganglia, even sympathetic or spinal, is the silver stain. Now we will talk about synapse. What is the synapse? Synapse means a site of signal transmission from one neuron to another neuron or to a muscle. As we said, that is the area or connecting area between two neurons or neuron and the muscle, where this contacting area or connecting area between them, it is the site of signal transmission. So when the signal transmitted from neuron to neuron or neuron to a muscle, the site between them that transmits this impulse is called a synapse. There is a type of synapses according to its contact. Axodendritic, from its name, this synapse between axon and dendrites, as we see. This is the axon of the nerve cell, and this is the dendrites of the another cell. So this synapse in between is between the axon and the dendrites, so it's called axodendritic. Another one is called the axosomatic, between the axon of one nerve cell and between the cell body or the soma of another cell body, so it's called the axosomatic. Another one is called the axonic, from its name, between two axons, between the axon of one cell body and the axon of another cell body, axonic. And the dendritodendritic, between dendrites of one cell to the dendrites of the another cell, so it's called the dendritodendritic. So according to the site of synaptic contact, there is four types of the synapses. Axodendritic synapse, between axon and dendrite. Axosomatic synapse, between axon and the soma of the cell body. Axonic, between two axons. Dendritodendritic, between two dendrites. Okay? There is another types, but according to the method of transmission between the two synapses. Transmission between the two synapses may be chemical, may be electrical, may be mixed electrochemical. The chemical transmission occurs by a release of neurotransmitters from the presynaptic to postsynaptic cells to generate the action potential, while the electrical occurs by movement of ions from one neuron to another neuron via the gap junction. This electrical synapse is less numerous than the chemical, but it's faster than the chemical. So again, we have three types of synapses according to the method of transmission between them. We have a chemical by release of a chemical substance, which is called the neurotransmitters, between the presynaptic cell and the postsynaptic cell to generate the action potential, and we'll discuss it separately. And there is another one is called electrical by the movement of ions from one neuron to another via the gap junction in between. This is type is less numerous and than the chemical but faster than the chemical. And if there is the two types of the transmission, chemical and electrical, is called electrochemical transmission. There is a third type of differentiation, or there is two types of synapses, but this time is according to the type of stimulation. Stimulation may be excitatory or inhibitory. Excitatory means the neurotransmitter hyperpolarizes the postsynaptic membrane, while inhibitory means that the neurotransmitter depolarizes the postsynaptic membrane. So again, we have two types of synapses, but this time is according to the type of stimulation. Stimulation may be excitatory or inhibitory. Excitatory means there is a hyperpolarization of the postsynaptic membrane, so it's called excitatory, or inhibitory means depolarization of the postsynaptic membrane, hence its name inhibitory. Okay? This is the chemical synapses that occurs by the neurotransmitter, which is a chemical substance. This is a nerve terminal of the axon and dendrites of another cell. This is an axon of one cell, and this is a dendrite of the another cell. This type of contact is called the axodendritic connection, okay, or axodendritic synapse. This area is called a synapse, formed from four parts. First, axon terminal, presynaptic membrane, presynaptic, which is the before of the synapse. This area is called the synapse. So the membrane before the synapse is called presynaptic membrane, and this area is called the synaptic cleft, and the membrane after the cleft is called the postsynaptic membrane. Axon terminal, it's a swelling containing mitochondria to produce ATP for this transmission, and the synaptic vesicles. These vesicles contain the neurotransmitters as acetylcholine. This is the presynaptic membrane, contains channels through which the neurotransmitter passes through the cleft, and there is a synaptic cleft which is 25 nanometer in width, traversed by a fine filaments to guide the neurotransmitter. This neurotransmitter arises from these vesicles, need a guide to know its way to the postsynaptic membrane. This guide is formed from fine filaments in the synaptic cleft. Finally, the postsynaptic membrane, which contains the receptors for the neurotransmitters, comes from the from the axon terminals. So again, this chemical synapse formed from four parts. Axon terminals contain mitochondria and the vesicles that contain neurotransmitters. Presynaptic membrane contains channels through which the neurotransmitter passes through the cleft, and then synaptic cleft, which is 25 nanometer in width and contain filaments that guide the neurotransmitters, and finally post-synaptic membrane that contain a receptor for the neurotransmitters. These four parts form the chemical synapse through which the impulse transmitted from this axon to these dendrites. This is the axon terminals by the electron microscope. This is a mitochondria, and this is the axon vesicles that contain neurotransmitters. This cleft is called synaptic cleft. This is the axon terminals contain circles. These circles is called the presynaptic vesicles. Presynaptic vessels, and this is the presynaptic membrane. This space is called the synaptic cleft, and this membrane is called the postsynaptic membrane. So this is the chemical synapse by electron microscope. Again, presented the vesicles, the presynaptic membrane, synaptic cleft, and postsynaptic membrane. Look at this video. This is the axon terminals, and this is the vesicles that contain the neurotransmitters, comes to the presynaptic membrane and passes through it to synaptic cleft, and then enters the postsynaptic membrane through these receptors, and the impulse is transmitted. Again, watch it. Okay. This is how is impulse passes from neuron to another neuron, also from neuron to another neuron. These dots like rings, this is the neurotransmitter. This is also neurotransmitter passes from the nerve to another nerve to pass the impulse, and from one nerve to another nerve. Neuroglia. Neuroglia, it is the supporting cells of the nervous system, CNS or PNS. So any supporting cell presenting in the nervous system is called the neuroglia. The characters of the neuroglia that it is 10 times more numerous than neurons. Neuroglia presented more than the nerve cells itself, 10 times more numerous than neurons. This neuroglia is not a nerve cell, so it don't conduct impulse or synapse. Okay? Special staining of the neuroglia is gold stain or silver stain. Okay? Neuroglia presented in the peripheral nervous system is two cells, satellite cell and Schwann cell, while that of the CNS it have four cells: ependymal cell, astrocyte, which is the largest one, microglia, and oligodendrocyte, which is the most numerous one. Again, neuroglia, which is supporting cell of the nervous system, 10 times presented more numerous than the neurons, which don't conduct impulse or a synapse. It has a special stain, which is a gold or silver staining. There is 6 types of cells, 2 in the peripheral nerves, satellite and Schwann, and 4 in the CNS, ependymal, microglia, astrocyte, oligodendrocyte. The largest one is the astrocyte, and the most numerous one is oligodendrocyte. Okay? This diagram presents the peripheral nervous system, which contains Schwann cell and satellite cells only, while CNS contains the four ones: oligodendrocytes between the nerves, ependymal cell, astrocyte, and the microglial cell. We will talk about each of these 6 cells separately. In this slide, note that there is a 4 nerve cells only: 1, 2, 3, 4. We know it by the cell body, while all of these dots represent the glial cell or neuroglial cell, as we said that the glial cells is 10 times more numerous than nerve cells. We have 4 nerve cells only, and all these dots represent the glial, which confirm that info that neuroglial cell is 10 times more numerous than the nerve cells. Let's talk about each cell of the neuroglial cell separately. Astrocyte is the largest one of the neuroglial cell, originated from the stem cells of the neuroglia. Neuroglia contain stem cells, which are called spongioblast. So any neuroglia originated from its stem cell, which is called spongioblast. As we know, blast means formating cell. There is two types of astrocyte, protoplasmic and fibrous. Protoplasmic presented in gray matter and fibrous presented in white matter. This cell, don't forget, is a C presented in the CNS only. So gray matter and white matter, it's part of the CNS. What is the function of astrocyte? Astrocyte responsible for support, nutrition, and insulator for synaptic contact and formation of scar in the CNS called gliosis. If there is a defect or degeneration in the CNS, as we know that nerve cells contain no centrioles, so it can't be divided or regeneration. So if there is injury occurs, the healing occurs by formation of scar. Scar formation occurs by aggregation of glial cell, mainly astrocyte. This aggregation called gliosis, like that of the connective tissue, which is called fibrosis. Astrocyte also responsible for maintaining the blood-brain barrier. Great. The cytoplasm of the astrocyte contain what's called the intermediate filaments. Intermediate filaments composed of glial fibrillary acidic proteins, which is the brown parts. Glial fibrillary acidic proteins contain it, which is the intermediate filaments presented in the cytoplasm of astrocyte. Blood-brain barrier, which is maintained by the astrocyte. It is a selective barrier, selective, not passing all the molecules or elements. It selects what it could be passed through it. Selective barrier between the blood and the brain. Why? To prevent the passage of toxic substance from blood to brain. This blood-brain barrier is formed by three structures. Endothelial cells of the capillaries joined by zona occludens, which is a type of tight junctions. Second, the basement membrane of this endothelial cell. Third, the feet of astrocyte or astrocyte processes that form a complete envelope around the capillaries. So again, the blood-brain barrier is a selective barrier between blood and brain to protect the brain from harmful or toxic substance in the blood. Formed from three structures: from inside endothelial cell, basement membrane of the endothelial cell, and the food processes of the astrocyte, which complete envelop the capillaries. 1, 2, 3. Okay. This is the blood-brain barrier. This is the blood-brain barrier. First, endothelial cell. Second, the basement membrane. Third, foods of the astrocyte. Again, endothelial cell, basement membrane, foci of the endothelial, foci of the astrocyte. Foot of astrocyte, basement membrane, endothelial cell. This is the blood-brain barrier. A second cell of the CNS is called oligodendrocyte, which is the most numerous one. Originated as usual from the stem cell of the neuroglia, which is called the spongioblast. This cell is presented in both gray matter and white matter. This is the oligodendrocyte, supports the cells, responsible for formation of myelin in the CNS. Each process rotates around one intranodal segment. So the cell responsible for formation of myelin in the peripheral nervous system is my Schwann cell, while that of the CNS is oligodendrocyte. So myelin forming cells. Two cells is called myelin forming cells, Schwann cell and oligodendrocyte, the two cells responsible for formation of myelin. What is the difference between them? My Schwann cell is a flat cell presented in the peripheral nervous system, while oligodendrocyte is a branched cell presented in the CNS. Myelin, Schwann cell, it forms a myelin for one intranodal segment, while oligodendrocyte forms a myelin for many intranodal segments. Neurolemma, it forms neurolemma, while oligodendrocyte, it doesn't form neurolemma. Role in nerve regeneration, Schwann cell has a role in nerve regeneration, while oligo has no role in nerve regeneration. So again, what is the difference between myelin forming cell? Schwann cell is a flat cell presented in the peripheral nervous system, forms a myelin for one nodal intranodal segment, and forms neurolemma with an important role in nerve regeneration, while oligodendrocyte is a branched cell presented in CNS, forms the myelin for many intranodal segments, which doesn't form a neurolemma and don't have a role in nerve regeneration. This is a Schwann cell presented in the peripheral nervous system, forms the myelination, while this is for one intranodal segment, while this oligodendrocyte forms the myelination for many intranodal segments in the CNS. Another cell from the neuroglia of the CNS is microglia. Microglia originated from blood monocyte. From this information, we know that it's a phagocytic cell. Why? Because the monocyte is the origin of macrophages. So, as microglia originated from blood monocyte, so it will have a phagocytic function. Let's continue and see. Microglia presented in both gray and white matter. Okay. Each structure is spindle in shape with central oval nuclei. Their processes are short and branching, stained vitally by trypan blue, and the function is phagocytosis. Hence, it is originated from blood monocyte. Again, microglia formed from blood monocyte, so its function is phagocytosis. This cell, presented in gray and white matter, presented as a small spindle-shaped cell with central oval nuclei, stained vitally by trypan blue. Another and last cell of the CNS neuroglia is the ependymal cell. Ependymal cell, it's a cubical epithelial cell, like that, lining the neural tube and the ventricles, and have a basal processes. These basal processes extend into the gray matter. They help or they have a cilia to help the movement of CNS, and they assist in the formation of CNS. So, roughly, ependymal cell, type of the neuroglial cell, cubical in shape, have a cilia from above to help in the movement and the formation of CNS, and have a basal process that extends to the gray matter. This cell lines the neural tube and the ventricles. The last cell of the neuroglial cells which presented in the peripheral nervous system is the satellite cell. Satellite cell is a small cell presented in the ganglia in the peripheral nervous system, and they form thin layer around the nerve cell body which forms a capsule cell. So again, be focused, presented in the ganglia in the BNS and forms a thin layer around the cell body, so it's called capsule cell. Okay, the function of satellite cell: protection, insulation, and nutrition as usual of the most of the neuroglial cells. So, Schwann cell and oligodendrocyte is the myelin forming cell. Ependymal cell, CSF and blood-brain barrier cubical in shape. Astrocyte, blood-brain barrier. Microglia responsible for phagocytosis. Okay. Nerve degeneration. What is the nerve degeneration? There is an injury in the nerve fiber. This is the axon, and this is the site of the nerve damage, and this is the fragments of the axons, although this is a fragments, and this is a droplets of myelin. What is the why it appears as a droplets? Because myelin is a fatty tube. Okay. Degeneration of nerve tissue. If there is a degeneration occurs in the nerve tissue, it differs according to its site. If it occurs in the CNS fibers, it will be permanent damage. But if it occurs in the peripheral nerve system, it will be followed by regeneration. This regeneration occurs by or occurs in 3 steps. After traumatic degeneration at the site of injury, there is a retrograde degeneration occurs in the cell body. Why it's called retrograde? Because the normal pathway is from cell body to the axon terminal. But if the pathway is reserved, it's called retrograde degeneration. While the other or distal part is called Wallerian degeneration. We will discuss it in details right now. Retrograde degeneration is the changes that occur in the cell body after injury of its axon. There is an injury occurs in the axon. What happened in the cell body? The actions or the that happen in the cell body, it's called the retrograde degeneration. What is these changes that occur? The cell itself, it will be swelled and withdraw its processes, so it becomes spherical. The nucleus became eccentric and pycnotic. The nether granules, it's break down into fine granules and disappear, which are called chromatolysis. Neurofibrils and the Golgi disappears. The remnant of this, all of this, finally, it will undergoes Wallerian degeneration. Again, Retrograde degeneration means changes occurs in the cell body after injury of the axon. What happened in the cell body? The cell itself withdraw its processes and became spherical and swell. Nucleus became eccentric and pycnotic. Nasal granules break down by chromatolysis. Neurofibrils and the Golgi disappears. Finally, the cell body degenerated and the axon undergoes Wallerian degeneration. This is the neurons and the axon, Schwann cell. There is the site of injury. Site of the injury leads to chromatolysis of the nasal bodies, and the nucleus became eccentric, and the cell body will swell, and then it will lose its processes. Wallerian degeneration, it is the degenerative changes that occur in the distal segment in the axon. What happened? The axon will undergo rapid degeneration in few days. The neurofibrils break down into fine granules, which finally disappear. Myelin sheath is fragmented into fat droplets, and the Schwann cell don't degenerate, but it proliferates to form a thick tube distal to the injury, which acts as a guide for the regenerating axon. Post-degenerating products from myelin sheath and axon will be removed by macrophage. So, if there is injury occurs in the nerve, there is a changes occurs in the cell body, and the changes occurs in the axon. The changes occurs in the cell body called the retrograde degeneration. The changes occurs in the axon is called Wallerian degeneration. What happened in the axon or Wallerian degeneration? The axon will undergoes rapid degeneration and the neurofibrils break down. Myelin sheath fragmented into fat droplets, and the degenerative products of both are removed by macrophages, while Schwann cell don't degenerate, but it is regenerate and proliferate to form a distal tube that act as a guide for the new regenerating axons. Okay? Okay. This is a Schwann cell, not degenerate, but it proliferate and increase in number to form a tube that act as a guide for the new regenerating axon. So let's summarize this degeneration. If there is injury occurs in the axon, there is a changes occurs in the cell body, and the changes occurs in the axon. Changes occurs in the cell body called retrograde degeneration. Changes that occur in the axon is called the Wallerian degeneration. What happened in the cell body? The cell will swell, nucleus became eccentric, nasal body disappears by chromatolysis, okay, and the cell became spherical and lost its processes. Golgi and the neurofibrils will disappear. The axon undergoes Wallerian degeneration, where it degenerate, myelin sheath is degenerated to fat droplets, then the both are phagocytosed by macrophage, while Schwann cell forming a proliferates and forming a tube that will be a guide for the new regenerating axon. Then the injury is healed and regenerating axon is formed. So what is the factor that may affect the regeneration of the nerve? Is the all nerves could be regenerated or not? There is a factors that affect this regeneration. What is these factors? Schwann cell presented or not? the cut type, partial or total, the infection, as it leads to formation of fibrous tissue that fills the gap between the two ends and prevent the healing from occurring. The distance between the two ends, small or large. So there is very four factors that will affect the regenerating process of the nerve fibers. Presence of Schwann cell, the type of the cut is partial or total, the distance between the two ends is small or large, and presence of infection or not, as the infection leads to formation of the fibrous tissue that fills the gap of the two between the two ends and prevent it from healing. Okay? Mechanism of regeneration. The neurolemma proliferate, forming the guide regenerating tube. Nerve cell resume its normal structure. The several branches grow at rate of 1.5 mm per day in human, and Schwann cell forms then a myelin sheath around the formed axon. Again, there is injury occurs here. How the regeneration occurs? The neurolemma proliferate, forming a guide tube. The nerve cells resume its normal structure after spherical in shape. Several branches grows at rate of 1.5 mm per day in human, and Schwann cell forms a myelin, and the regeneration occurs, and the nerve resume its normal structure again. That's it today, and thank you for your listening, and have a nice day, guys. Thank you.